Welcome everyone to my channel BioWorld once again. So again with the same chapter genetics after monohybrid cross and incomplete dominance. The next important part of this chapter is codominance. From the word itself you understand codominance. That means the in-between dominating trait. So the definition I wrote here, you can see over here, a type of inheritance in which two alleles of the same gene are expressed separately to yield different traits in an individual. That means the gene is same, the two alleles of a particular character is expressed in a different way. And the most important example is blood group in human. Now we know we have come across ABO blood group system in human, where we have blood group A, blood group B, blood group AB and blood group O. And we have categorized them on the basis of the antigen, that is a specific type of protein which is found on the surface of RBC. So ABO blood group is one of the most important example of codominance as well as multiple allele. Now human is a diploid organism. What is the meaning of diploid? That means each character carries two alleles. A gene has two allele, might be one dominating, other recessive, or both recessive or both dominating. But in what is the exception case in blood group, that is human blood group, that a particular character has three alleles. What is they? They are IA, IB and small I. You can see on the board IA, IB and small I. Now, ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cell has some sugar polymers that comes out from its surface and it is controlled by a particular gene. And this gene has three alleles, that is IA, IB and I. Now IA and IB, they produces different forms of sugar, whereas small I, it do not produces any kind of sugar. So because that is the nature of the diploid organism, that two of the alleles would be producing the character trait. That is the example. So what happened here? So as humans are diploid, each person possesses any two of the three alleles. That is IA and IB are completely dominant over small I because it do not produces any sugar. So IA and IB, they are dominating over small I. Now let's see in human blood group how the blood group is produced, or you can say how the genes or alleles they produces by combination of the alleles. That is, the offspring blood group is defined in what way? So I have written here alleles of parent 1, alleles of parent 2, genotype when they are combined and blood group when they are produced. Now see, from parent 1 IA, that is A producing allele, that is sugar producing A1, allele from parent 2 also A. So both are same type, that is combination when it happens, it comes out IA. The blood group which is produced is A. In the second case, it has been written IA from parent 1, IB from parent 2. Combination of genotype in that offspring is IA, IB. That means both A and B will be producing their sugar respectively, and the blood group of that offspring will be AB. Next one is IA and small I. As we know, small I do not produces any sugar, so IA is dominating over small I. So the combination is IA, small I, and the blood group will be A in that offspring. Next one is IB, IA. Again the combination is IA and B, and the blood group would be AB. Next is IB. IB here is very simple. Both B, hence the blood group would be B. IB small I. Again here, as I do not produces any of the sugar, hence B is dominating on it. So the blood group would be produced in that particular human, B. But when both I and small I, they come up from the parent, when the parent combination is small I and small I, definitely there is no sugar production, and that combination is I, and the blood group of that person is called O blood group, as we have known that O has no antigen, and hence it is also considered as universal donor. So in this way, the blood group which produces three different category of alleles differently here, hence it is known as codominance in one particular trait, and also ABO blood group is an example of multiple alleles. Since in any individual only two alleles can be present, multiple alleles can be found only when population studies are made. So, in a diploid organism, only two alleles are produced or are present in a particular trait. So, a single gene product may produce more than one effect. Okay, so this way, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of particular phenotype. So we need to understand how the alleles and the gene, they are influenced, and how the phenotype, they are influenced by that particular allele of the gene for a particular trait. So in this way, human blood group is an example of codominance, which is alleles of the same gene which could be expressed separately to produce different character in an individual. So this was codominance. Please go through it. Do like it and share and subscribe my channel. Thank you for watching it.